So let's quickly have a look at cytotoxic chemotherapeutic agents. So these are drugs that we use to kill off cancerous cells. Now, remember with cancer that these cells are rapidly dividing and no longer listen to the signals telling the cell to stop dividing. So these drug therapies target specifically rapidly dividing cells. Now, we try and aim these drugs at rapidly dividing cancerous cells, but unfortunately you'll find that these drugs also tend to target rapidly dividing normal cells of the body, such as the bone marrow, such as those of hair, and also such as those of the stomach or gut. Which means some of the side effects that patients may exhibit include side effects of affecting these particular organ systems, such as alopecia, which is hair loss, gastrointestinal issues, and also problems with blood levels, whether that be white blood cells, leukocytopenia, red blood cells, anemia, or even platelets, which is thrombocytopenia. Now, let's have a look at some of the classes that we could classify these cytotoxic drugs into. So, you'll find that there are 4 broad classes, and then we can look at some of the subdivisions of chemotherapeutic agents within those broader classes. So first, there are what we call cell cycle specific, agents, and there are cell cycle non-specific agents. Now, I'm just going to stop here for 1 second because we need to talk about the cell cycle. So remember, the whole point of the cell cycle is it's a series of events that occur within a cell that prepares that cell for division. Now think about what a cell needs to do in order to prepare itself to split into 2. Well, it needs to increase the amount of contents it has, so increase the amount of proteins, increase the amount of metabolites and so forth within that cell to prepare it to split in 2. It also needs to copy or duplicate, double the amount of DNA that's present. And then once that's happened, it needs all the right ingredients and components to be able to split that cell apart. This is the cell cycle. Now, we can have chemotherapeutic agents that act during certain phases of the cell cycle, and these are the cell cycle specific. So let's write down during cell cycle phases. Or you can have chemotherapeutic agents that don't need to be, that can have their effect while the cell isn't within any of these phases of the cell cycle. So basically when the cell is at rest, or when the cell cycle is at rest. There are 2 other broad categories. We have tissue specific agents, and we also have miscellaneous. So this is just a category in which the therapeutic agents that fit within don't actually fit within these other categories. Alright, so let's first start off by looking at the cell cycle specific agents. So the chemotherapeutic agents that need to act during certain phases of the cell cycle. That means that they need to be given at particular times to the patient. So there are 3 individual categories that I want to place within here. So first of all, we have the antimetabolites. Now, when a cell is undergoing the cell cycle or cellular division, like I said, it needs to basically double the amount of stuff that it has. Now, that means it needs to double the amount of DNA that it has as well. Now, when it does this, it needs to take certain metabolic products or molecules for this process. Now, when we make DNA, you know that DNA is that double-stranded, I'll draw it up. You've got double-stranded DNA, like that. And you know that there are certain what we call base pairs that comprise the backbone of this DNA. So you've got A's, C's, T's, and G's. And A's will always bind to T's, C's will always bind to G's, and so forth. And this is all throughout our DNA, all throughout our 3 billion base pairs of our DNA. So when we need to make, so I've got one strand here, one double-stranded bit of DNA here, I need to make another strand. How do we do it? Well, we split it apart, and then we copy both strands, and then we have double. But that means in order to do so, when I split this strand apart, that means so I've split the strand apart, like so. I'm going to have an A here, for example, and there was the T there that it was attached to, but now I need to make a copy of this strand, which means I need to pull some metabolites in, or some molecules in, to make that T. So I can start the new strand, which means the other new strand here will need an A. Okay? But where do I get these T's and A's and C's and G's from? Well, we get them from the food that we eat. So when you eat a steak, for example, and you mechanically digest it, and then you chemically digest it, and you break it up into its micronutrients, you break it up into nucleotides. And we reuse these nucleotides in processes like this. So, as an example, one of the chemotherapeutic antimetabolites that we can use include one called 5-fluorouracil, and what 5-fluorouracil does is it takes the place basically of thymidine. So remember, you have A, C, T, and G. That's thymine or thymidine. And so 5-fluorouracil just takes its place. So when it's time to pull upon or call upon a thymidine, well, that takes its place, and then it basically renders this DNA molecule inoperable, unusable. So it doesn't participate in that synthesis, which means when these rapidly dividing cancer cells are trying to make new copies of DNA, 5-fluorouracil will not allow for this to happen. So this is a cell cycle specific medication because it needs to be involved in the synthesis phase of DNA replication. So, what's another cell cycle specific agent? We have anti-tumor agents. So these anti-tumor agents are actually anti-tumor antibiotics. And they're derived from microorganisms. So for example, streptomyces is a microorganism in which one of these anti-tumor antibiotics is derived from. Streptomyces, and this specific type of anti-tumor antibiotic is called doxorubicin. Now, what doxorubicin does is that it plays a role by intercalating into the DNA. What's intercalating mean? Well, you know how I was just showing you before how that new DNA molecule needs to pull upon A's and C's and T's and G's to make a new strand of DNA? Well, what doxorubicin does is when an A comes along and then a G comes along, for example, it pops in between. So it intercalates, it sits between base pairs of the DNA. And again, this makes the DNA unable to synthesize. So that's doxorubicin. And then the last type of cell cycle specific agents are the plant alkaloids. Now the plant alkaloids are derived from plants. So for example, one plant is the mayapple. And what you'll find is that these types of plant alkaloids, such as those isolated from the mayapple, which is called etopside, the way it works is that it stops the spindles. So what are the spindles? The mitotic spindles. So in the cell cycle, once you've doubled up all that DNA, once you've doubled the chromosomes, you need to pull the cell apart. which means you need to pull those chromosomes apart. And there's certain subcellular structures that do this, which are called mitotic spindles. And the top side, from the mayapple, stops these mitotic spindles from being able to pull the chromosomes apart. And so again, this is important in the mitotic phase of the cell cycle. So you can see why these three classes, antimetabolites, anti-tumor antibiotics, and plant alkaloids, are all known as cell cycle specific agents. 5-fluorouracil takes the place of thymidine, so it can't synthesize DNA. Doxorubicin intercalates between those base pairs. Again, you can't synthesize the DNA. And etopside stops the mitotic spindles from being able to pull the chromosomes apart. What about the cell cycle non-specific? So this means that these drugs can be given pretty much at any time, and it can act even while the cell cycle is at rest, so not within the cell cycle. So the one class I want to talk about are the alkylating agents. Now what this means is, alkylating agents means that it forms an alkyl group to the DNA. It adds an alkyl group, and you don't really need to know too much about what that refers to unless you're a chemistry major. But one specific type of alkylating agent I want to talk about, because there's many different types. There's nitrogenous based ones, metal based ones, for example. I want to talk about a metal based alkylating agent, which is called cisplatin. Cisplatin. And that platin actually stands for platinum. And this drug was actually first identified back in the 1960s, when they used to do a lot of their basic research. And one researcher called Dr. Rosenberg in the 1960s was doing a number of experiments growing up bacteria and trying to grow that bacteria between electrical currents and seeing what electrical currents did to the cell cycle. And this doctor found that this particular electrical current stopped the cell cycle from working or happening. And he did it again and realized that it wasn't the electricity per se that was doing it, but the electrodes, because the electrodes were made out of platinum. And as this electrical current was being put across, some of the platinum was dropping into the solution and forming cisplatinum. And it was cisplatinum that was stopping the cell cycle. Okay, now how does cisplatinum work? Well, it forms crosslinks into the DNA. Crosslinks in DNA. That's different to intercalating in the DNA like doxorubicin. Cross-linking means that it forms attachments between the two strands or within a particular strand, which again, this can happen at any time. Remember, doxorubicin is intercalating. So when the new base pairs are being laid down during the synthesis phase, doxorubicin can jump in and stop it from happening. But crosslinks can happen even when the DNA isn't being synthesized. It will just jump into the DNA and form these crosslinks. That's why cisplatin is a non-specific agent. All right, let's move to the tissue-specific agents. These are hormonal agents. So some cancers are heavily dependent upon hormones within the body. For example, breast cancer and prostate cancer are both heavily dependent upon hormones and hormone level within the body. So if we take breast cancer, for example, what you'll find, so I'll write breast cancer. Breast cancer is highly dependent upon estrogen. So you can have certain medications, such as those which are called SERM. So SERM is selective estrogen receptor modulators. Selective estrogen, let's write it down. Selective, now because we're Australian, we put the O before the E. Estrogen receptor modulation. And basically these particular chemotherapeutic agents, what do? They act sort of like antagonists, but not really. They modulate the receptors. Okay? They modulate, in this case, estrogen receptors. Now, the reason why I have to say modulation is because some receptors it upregulates and makes that tissue type more sensitive to estrogen. But other tissues, such as those of the breast, it actually makes it less sensitive and acts like an antagonist. Hence why these drugs, SEM medications, are used for tissue-specific or as tissue-specific agents. One type of estrogen receptor antagonist, in a way, is tamoxifen. And basically works via this mechanism of action. Okay, last type, miscellaneous. Now, these types do not fit within these other categories, and there's many different types of miscellaneous chemotherapies, but I'm just going to talk about one, which is the monoclonal antibodies. So, this is one of the most specific ways to target cancer. And the reason why is because it utilizes our own immune system, or the patient's own immune system. The way it works is, you know that all proteins in the body have little flags on them, okay? Or tissue cells, they all have little flags on them. These flags are called antigens. And these antigens basically just present that cell as saying, I belong to you, or I do not belong to you, okay? Now, our immune system will target antigens that tend to not belong to us. Now, sometimes it does target antigens that do belong to us, and this is known as an autoimmune reaction. But that also means that cancerous cells have cancer-specific antigens, cancer-specific flags, waving, telling the body, I shouldn't be here. And so we can create, similar to vaccines, a means of targeting these antigens by stimulating the body's own immune system to create T cells that attack and target, or target and attack, specific cancer cells by the antigens that they are presenting. Okay? So monoclonal antibodies are antigen specific. Okay? And this can include certain types of cancer like vaccines. And that's where I'll finish it. So hopefully that all made sense, looking at cell cycle specific agents, cell cycle non-specific agents, tissue specific and miscellaneous when it comes to looking at cytotoxic chemotherapeutic agents for cancer.